As the world finds its way forward, people never stop needing ways to conquer disease, ways to live with HIV, ways back from cancer. So you can count on Janssen to keep producing the medicines you need while we continue work to help end the pandemic. Janssen is the pharmaceutical companies of Johnson & Johnson. We never stop working toward a future where disease is a thing of the past.
Hello, good morning, good afternoon, good evening. My name is Louise Thwaites, and I am the moderator for this first session of our special Spotlight event today. It is my pleasure to be opening this very first session of the event. I'd like to thank our exclusive sponsor, Janssen, for this session. Before we go on with our first speakers, however, though, I would like to invite Evangelos, our program chair, who's going to give the opening remarks.
Thank you very much. Good morning, everybody. My name is Evangelos Giamarellos. I'm professor of internal medicine and infectious diseases at the University of Athens. And it's a really honor for me to be the program chair of this Spotlight event of the Global Sepsis Alliance. And actually, our idea is that for all of us who are participating and who are attending and who are interested in the field of sepsis, it is a great opportunity because the pandemic and actually the level of awareness, of the widespread awareness and societal awareness about what a lethal infection is generating for us the path to communicate to everybody, not just to physicians, how important sepsis is, and this is the top killer. With this in mind, we have drafted a program which goes from the reaction of the host to a lethal infection, the prompt diagnostics, and the best therapeutic approaches with as much as possible to provide as many information as possible within 9 to 10 hours in order to acquaint you with the new developments in the field. So I would like to pass to Louise, the chair for the first session, and we are wishing to you a really enjoyable Spotlight event. Thank you very much.
Thank you, Evangelos. So we're about to go over to our first speaker. I'd just remind you that please put questions into the chat box. The first talk is actually going to be pre-recorded, so we can't have questions on that one, but we're really fortunate that all the other speakers in this session are here live and will be able to answer questions. So please do participate like that. So our first speaker is Peter Hotez. He is the Dean of the National School of Tropical Medicine and Professor of Pediatrics, Molecular Virology and Microbiology at Baylor College of Medicine. He's also the co-director of the Texas Children's Center for Vaccine Development and Texas Children's Hospital Endowed Chair of Tropical Pediatrics. He's university professor at Baylor University, fellow in disease poverty at James A. Baker III Institute for Public Policy, senior fellow at the Scowcroft Institute of International Affairs at Texas A&M University, a faculty fellow at the Hagler Institute for Advanced Studies, and the health policy scholar at Baylor Center for Medical Ethics and Health Policy. And he's going to be talking today on the role of science and novel technologies to combat 21st century health threats.
Thank you for the opportunity for me to say a few words for the World Sepsis Congress and the Global Sepsis Alliance, and congratulations on all your important work, and look forward to hearing more about your findings regarding the therapeutic and diagnostic approaches to COVID-19. As we approach our third year now of this horrible pandemic, we're realizing the loss of life caused by COVID-19. While the official estimates are between 5 to 6 million, many, such as the Institute for Health Metrics and Evaluation, put it closer to 20 million and maybe 4 or 5 million deaths in India alone. And nations such as the United States and Russia, we're looking at easily over 1 million deaths. So horrific human tragedy. In part because we've failed to rapidly accelerate our ability to vaccinate the world, especially the world's low and middle income countries, and to provide therapeutics for low and middle income countries. And to me, this vaccine inequality remains one of our great failures. I think, the COVAX sharing facility was an important organization for sharing vaccine doses. I think in many ways there was some upstream science policy failures, that there was so much emphasis on speed and innovation. And we did get some interesting vaccines, mRNA vaccines, adenovirus vectored vaccines, DNA vaccines, particle vaccines. But of course, as any engineer might tell you, when you rely on a brand new technology, there's a learning curve before you can go from 0 to the estimated 9 to 10 billion doses needed for the world's low and middle income countries. And there wasn't that ability to rapidly scale a brand new technology. Our approach has been to focus on the vaccine producers that are already in place in low and middle income countries, in places such as India, Indonesia, Bangladesh, Vietnam, Brazil, Argentina, Cuba, which already have the ability to make their own vaccines using microbial fermentation and yeast, recombinant protein vaccines. And this was our approach to be able to use that system that's already been in place to produce recombinant protein hepatitis B vaccine. For example, a low-cost vaccine, a vegan technology, no animal cells and human cells. That was been our approach for our parasitic disease vaccines, coronavirus vaccines for SARS and MERS, and now COVID-19 vaccines. And that's been in place now, and we transferred the technology with no patents and involved, because, as I often like to say, when your house is on fire and you can make one phone call, you don't call the patent attorney, you call the fire department. So we hope to be the fire department. So our Texas Children's Hospital Center for Vaccine Development did that technology transfer. It's now been scaled in India to 300 million doses with plans for over 1 billion. It's been released for emergency use authorization. It's been given to almost 30 million adolescent school children in India, and now it's just been approved for the 5 to 11-year-olds. So the hope is this can start, projects such as this can start addressing the terrible vaccine inequalities. I think equally important is we have to address a very aggressive and dangerous anti-vaccine movement that I now call an anti-science movement. And I don't even call it misinformation or disinformation anymore. I call it anti-science aggression. The numbers are quite chilling. So for instance, here in the United States, since May 1, 2021, that's the date last year, when the Biden White House announced that any American who wants to get vaccinated will have access to an effective COVID vaccine, we had a large segment of the population refuse to take a vaccine. So after May 1, 200,000 additional unvaccinated Americans needlessly lost their lives because they refused to get vaccinated out of defiance, out of refusal. Actually, I consider them victims of anti-vaccine, anti-science aggression, which has become a dominant force in the US. And now, unfortunately, it's accelerating into Canada, into Western Europe, the Nordic countries, elsewhere. And so we have to be cognizant that now this is a force to be reckoned with in the world's low and middle income countries. So it's very interesting. I've devoted my life to being a vaccine scientist and to making vaccines for poverty-related neglected diseases and now COVID-19. But increasingly, I recognize to really pursue science for humanitarian goals, it also means combating the anti-science. And As some might know, I have 4 adult kids, including Rachel, who has autism and intellectual disabilities. And a few years ago, I wrote a book called Vaccines Did Not Cause Rachel's Autism, to counter what the anti-vaccine groups were saying. And that made me a public enemy #1 or 2, but it also gave me some insight and expertise into these anti-vaccines, anti-science movements, and they are no longer mom and pop organizations. They're well-funded, well-organized entities that's causing a lot of damage. And now we're seeing its impact on causing people to refuse COVID-19 vaccinations, causing terrific losses of life. And I worry now that it's not going to stop at COVID-19 vaccines. We're already seeing spillover now with convincing parents not to vaccinate their kids against ancient childhood scourges such as measles. And so I worry that we're going to be looking at a drop-off in global vaccinations for children. And I also worry about its impact on other therapeutic interventions. We've already seen how the anti-science lobby aggressively promoted spectacular cures that did not work, such as hydroxychloroquine and ivermectin, and I worry this is going to affect the therapeutic realm as well. So I hate to end on that sort of sour note, but just keep in mind that you're doing important work, and your work is going to be critical not only for promoting good science and new therapeutics and diagnostics for COVID-19 and of course for sepsis, but also combating the anti-science. And this is going to be our new challenge for our profession. So thank you for the opportunity to address you. Good luck on your Congress, and I'm very excited to be hearing about your progress. Thanks.
Thank you. Thank you, Peter, in your absence. So as I said, unfortunately, we can't take questions for that talk right now, but that does give us a little bit more time for the other speakers in this session. So without further ado, we will go on to our next talk. This is by Direk Limmathurotsakul, who's from Mahidol Oxford Tropical Research Unit, where he is head of microbiology. He's also the associate professor at the Faculty of Tropical Medicine at Mahidol University in Bangkok in Thailand. Direk special interests are many infectious diseases, including melioidosis and also antimicrobial resistance. And he's going to be talking to us this morning, this evening, wherever you are in the world, on what is missing in surveillance of AMR bacteria in hospitals in low middle income countries and high income countries. So without further ado, over to you, Direk.
Thank you very much, Louise, and everyone and that coming for today and Global Sepsis Alliance. And I also work in the sepsis, so I will time my talk about the AMR surveillance and the sepsis as well, and what I personally feel that is still missing in the AMR surveillance. So, first of all, you might have seen what is the global burden of the AMR. You might have seen the big paper about what is the global burden of sepsis, and the next one is the global burden of the AMR. And if you follow the publications that come out, that is about 5 million deaths associated with the bacterial AMR. This is based on the additive scenario, that is AMR versus non-infection. And it could be about 1.3 million deaths worldwide based on the replacement model of the AMR versus AMS, or antimicrobial susceptible infection. The key that I think is missing from the big paper that they will have to go on is actually then how many die in each country. How about in your country? How about in each low and middle income countries? I think among high income countries, you have only in the Europe, UK, and US that each Public Health England, CDC, or ECDC is doing that. But they're not even in China, Japan, or upper middle income countries. And then we have to think in the same way that every country nowadays publishes how many die of COVID each day, each year. You have seen the raw data. You have seen some model estimated how many underestimated deaths from COVID in the last year. I think in the same way, the AMR should do the same thing in both all high-income countries and in low and middle-income countries. Every country should show their raw data, how many die of or die associated with, based on the raw data, and then the estimated data can come further. Next, I think the key things that coming highlight nowadays is in the WHO Sustainable Development Goal. They talk about the indicators compared between the low middle income countries versus high income countries. The important one, if you go to the link to the website, they talk about the proportion of patients with bloodstream infection caused by E. coli that are resistant to third generation cephalosporins, let's say 3TCR-EC, by country income level. The graph shows that the proportion is much higher in low and middle income countries and lower in the high income countries. And this parameter has been added into the SDGs parameter that we should look into it further, but they also highlight that these parameters need further investigations and why it happened. And I think many people have reasons about it, but we still don't know whether this difference of proportion is really the best marker for the bigger burden in that country or not. I will keep that in mind a little bit and we will look forward. I think the key things that I work into it, and it's related to the sepsis as well, because the guideline of the surveillance sepsis is that everyone who presents with sepsis should get the blood culture tested immediately, or if you cannot wait for the antibiotics, it should be immediately after antibiotics given. But then in every country, you have issues about underutilized of blood culture. Even in the high income countries, papers published from Europe, US still shows that blood culture are not using 100% among the patients who should get the blood culture. We work on it. We publish some issues about the impact of the low blood culture usage on the proportions and incidence rates of the AMR. And I'm going to talk a little bit about that work so that you can see what we work on it. In the reality, the blood culture even in the upper middle income country like a Thailand, if we look into the whole data, we have the blood culture on the first day. And then if the patients are not improving, we might repeat the blood culture before we switch the antibiotics. So we show the data of the first blood culture and repeated blood culture. And we took whole data into the modeling analysis. In the scenarios that if we take all of the data in, what our AMR report would look like. And then if we reduce the amount of the data from the first blood culture to be less and less and less, if we use only half of the first blood culture data, a quarter of the first blood culture data, or only just 10% of the first blood culture data, how our AMR report would look like. We kept all of the repeated blood culture data so that it can represent the delayed blood culture. Why we did this? Because there are a lot of talk about in the low middle income countries that patients present with sepsis, ceftriaxone. 2 days not better, ceftazidime, metronidazole, 5 days not better, okay, switch to carbapenem. 7 days, don't die and don't improve, 1 hemoculture bottle. We don't have much data to support that, but it's coming. But that is the scenarios why we took it into this modeling. So these figures will show you the data from a big tertiary care hospital in Thailand, 1,200 bed hospital. And if we use the whole hospital data of both hospital admission data and microbiology laboratory data into the analysis, we present you for the third generation cephalosporin resistant E. coli. And then you see that the total or overall proportion of 3TCR-EC is about 44%. If we split it into the community origin and hospital origin based on the WHO class, obviously the hospital origin will be higher and community origin would be lower. incidence rate in the very old method that per 100,000 population, and then the new parameters per 100,000 tested patients. But if we do less and less and less first blood culture, but keep the data of the repeat blood culture to represent the delay blood culture, how would it look like? If we do less and less and less first blood culture, the proportion of 3-GCIEC keep rising and rising because it represents the delayed blood culture. It represents the AMR from the patients who fail first empirical antibiotics. Incidence rate, on the other hand, keep declining because you have less culture positive patients. Incidence rate per 100,000 tested patients didn't change much. And if you took into the criteria of community origin and hospital origin, you will see that the proportion among community origin doesn't change much, but the accuracy or precision, you can see that is lower and lower by the bigger of 95% confidence interval. But the proportion among hospital origin bacteremia, culture positive for E. coli, the proportion of 3-GCIEC will keep rising. This is what you would observe if you do less and less blood culture, or mean the impact of the low blood culture utilization. And this could be one of the reasons that I show in the SDG parameter that low middle income countries had higher proportion of 3-GCIEC. That could be because in low middle income countries, one of the reasons they use less blood culture. The other parameter that is quite important is blood culture utilization rate. And this parameter also available in the Cassini papers for the whole Europe and in the WHO studies. And I think that it's good parameters, but cannot be specific among community origin, hospital origin. It cannot be specific about the type of the patients, or we call case mix. Some hospital might need to do more blood culture because we have more sepsis patients. We have more community-acquired sepsis patients. Some hospital might have less community-acquired sepsis patients, so they use less blood culture per 1,000 patient days. So that's why another parameter has been proposed, which quite long name, proportion of patients having blood culture taken on the day or within 1 day after antibiotic has been started. So that parameter can be helpful if people will do PPS, point prevalence survey, or review the chart of the whole sepsis patients. We propose that. And the other thing that it could be quite helpful among the AMR surveillance is that if you stratify the proportion of the AMR, and this is shown as a proportion of 3-GCIEC among patients with blood culture positive for E. coli. If you split it on the proportion of 3-GCIEC among patients who get blood culture immediately before or 1 day after antibiotic has started, or among the one who delayed antibiotics, you can see that the proportion of 3-GCIEC is much different. And that's why we think that in the surveillance of the AMR, maybe in hospital in both high-income countries and low-income countries, if possible, they should stratify by exposure to empirical antibiotics as well. And that would give us a bigger picture, a better idea about the proportion of AMR in different scenarios. The other thing that I think is quite important is how many days or weeks you need to generate your local antibiogram or cumulative AMR report. And even in the high-income countries, I heard that it could be weeks or months. And I think the key that we propose is, can we speed this up? And it has been generated for many low-middle income countries. Every country, every hospital has to do it every year. So we generate an application called AMAS, and we make the program open access. It's easy to use. It works offline. We collaborate with WhoNet. And if you are using WhoNet, now you can download the new version of the WhoNet that can call this application as well. The paper has been published. And the concept is that you don't want to transfer your local data of microbiology laboratory and hospital admission data to other persons, and you might transfer to the national authority and they send you the report many months later. But the key here is that you can just download the application to your own computer And many hospitals might use a different name of the variables. Some hospitals use the word sex, some hospitals use the word gender, some hospitals use the word male, female, some hospitals use 0 and 1 to represent male and female, for example. And that's why we use the concept of the dictionary in the Excel format. We don't ask you to be able to understand stat program like a data, SAS, or R. You should be able to use Excel and then put everything into the same folder, work on dictionary maybe 1 hour in the first time, and you just double click the application, and then the application will import the data from the Excel into the program, analyze, deduplicate, and make the PDF report for you. And then when you have the PDF report and summary Excel file, you can share that report to your national authority, international. You can even make it open access. And you can add the mortality data into it. If you have in-hospital mortality data, it will also come up the report. And that's the data that come back to the slide one that I think is so important in the future. And the key is that you should make AMR surveillance report or antibiogram open access. We did it, we show it, we make it in the fixed share, and we should acknowledge them. I think that to make all AMR surveillance report open access, that's a goal that we should aim for the whole world, both high-income countries and low-income countries. So, in summary, I think what is the missing? 1, how many die of AMR per each country, both raw data and estimated data. We should make all AMR surveillance report open access from every hospital, and that should be from both high-income and low-income countries. Mortality data at the moment is not in the AMR surveillance report. I think it should add in blood culture utilization rate. It's only in a few high-income countries nowadays that reported it. And I think every AMR surveillance report or every cumulative antibiogram, it should come up together as well. I think even if we work in the sepsis, what is the proportion of sepsis patients that get blood culture prior to the antibiotics? I think every country should report that as well. And then we should split or stratify the AMR report by the exposure of the antibiotic as well. I think the key main 5 things that I want to say, but then there's still many other things like a case-based surveillance, syndrome-based surveillance, or sepsis-based surveillance. Because when we work on the EMR, when we generate antibiogram report, we want it to have an impact on how we use antibiotics. But if we don't have the case-based or syndrome-based, it's quite difficult to convince clinicians to adjust or change their way to use of antibiotics based on the antibiogram. I think that's all for me. And I would like to say that, sorry, I forget to say that most of my study was funded by Wellcome Trust. So the committee within the CETRIC of the Wellcome Trust as well, they focus on the surveillance epidemiology of drug resistance. Over. Thank you very much.
Thank you very much, Direk. That was a really interesting talk, and I think a really nice one to follow on with. Talking about blood cultures is going to lead really nicely onto our other speakers who are going to be talking about some newer rapid diagnostics and newer techniques. But I think starting with the basics and blood cultures is really nice. I'm just looking at the questions here now. There is a question from me to start off with. You talked about the paucity really of data about sepsis and AMR generally. You've given us some examples, but are there any countries or systems that are working very well, or any examples where deaths and infections and everything is being reported nicely and could be a model for other countries? Are you aware of any of these systems?
At the moment, I think that only the ECDC, US, and Public Health England, UK, that do it systematically by the government. I think for the other countries, I think only Thailand that have quite a big data and the research on the mortality. I think the GRAM project of the University of Oxford that work on the global burden of AMR, pull together the data in as much as possible. I think that it's for the research, but I feel that follow up for the COVID, if the COVID organization is going to switch the work, I think they should switch to sepsis and the AMR. They should report how many die of sepsis and how many die of AMR yearly based on the routine data. That's what I think. So to answer in short, no, not many. And then I think we have organization in every country in the world working on that COVID. I think we should switch it further for the AMR and sepsis.
Great. Thank you. There is a question in the chat here. A substantial proportion of patients with COVID-19 develop secondary bacterial and fungal infections. They're asking, in general, what are the main infections, main secondary infections, I think, leading to death by continence? Are you aware of secondary infections in COVID? Is there any data available on, I guess, on the AMR surveillance generally?
Yeah, there are. I have my students working on the data in Indonesia. I crossed some reviews about it a bit. I think that the MRSA stuff, MRSA and E. coli, Cinetobacter are quite common, Klebsiella pneumoniae as well, and the fungal, both from the Candida albicans and non-albicans are popping up, and I think the new one from the India as well. But the data are not systematically reported. I think mostly are the what you call piecemeal researches from multiple areas, and the proportion of the AMR in some reports were higher among the COVID patients. Some reports didn't see the change of the proportion and rates. And I think the key is about the case mix, the severity of the COVID patients, steroid usage, and the IPC that they are implemented into the COVID ward as well. That make the difference we are observing. So that's what I think.
Great. Thank you very much, Direk. Well, I think we'll just have if you can answer very quickly the last question, and then we'll move on. Someone's just said, why do many doctors treat COVID with antibiotics?
I think that it's a combination. I think when we talk about behavior and antimicrobial stewardship, I think that the future is about the behavior of doctors, why they are following the norms and their local guidelines, and they heard a lot about the secondary bacterial infection. And then I think that the people worry or concerned about that, whether the secondary bacterial infections already from the start or after that. And I think that more and more data would come out, not work on it yet, but that doctors tend to prescribe antibiotics based on norm of what people among them normally do, rather than about the guideline or what they feel appropriate, because it's safer for them and the patient satisfaction is more important than the guideline in general. But I think more research have to work on it. But my update, diagnostic stewardship, highlight this issue. And I'm quite sure that antimicrobial stewardship is the same thing for that. Over.
Okay. Thank you. Thank you. And with that, we will move on to our next speaker, Eran Zahavy. Eran is originally a PhD in chemistry and biophysics from the Hebrew University and a postdoc from the University of Texas in Austin. But more recently, he's been the head of innovation of Israel's Institution for Biologic Research. He's worked part-time at Adaltis and now is CTO full-time for the bio division of BATM. He is going to be talking this morning about rapid multiplex diagnostic evaluation for sepsis using isothermal rolling circle amplification. So over to you, Eran.
So let me first thank you all for the meeting and relate to the previous speakers like Peter and Direk about the notions of the anti-scientist movement in the world. I think this is quite severe, and I really felt this notion during my position in IIBR during COVID time, where I had to face a lot of very weird and anti-scientific ideas and problem about this diagnostic and also therapeutics. So here we are in Adaltis and BATM and Adore company, in all the companies that we are holding and advancing. We are absolutely 100% pursuing a scientific solution to diagnostics issues, and mainly in infectious disease, not only in sepsis, in multirespiratory disease, in tuberculosis as well, in meningitis and other type of disease. So this is what we are doing. And here we want to present our solution to the sepsis issue. Adore is a company that is holding by Batam and Adaltis, which is dealing with diagnostic, IVD diagnostic, funded in 2016, and we are producing a point of care solution to the infectious disease community. We are holding a very big lab in Rome, in Adaltis facility, which was Merck Serono Diagnostic, and also in Rehovot, Israel, where we are developing a unique solution for this problem. One of the disease that we want to face is a sepsis, of course, which, of course, it's a very complex and acute condition disease, which require a very rapid diagnostics and treatment. And this is, and comparing to the old gold standard of blood culture, which takes a lot of time and a lot of experience and subjective analysis, and cause a very high burden in hospitalization and expenses. So what we are offering is to face directly the sepsis diagnostic challenge. And for this, it's also a variety of different bacteria that can cause sepsis, which you can see the list here and you all know them. Also, as my previous speakers was talking about AMR, antibiotic-resistant bacteria, which we need to face, which are a subspecies of the common bacteria, and also deal with viral condition, which might cause also sepsis as a septic shock. So we want to introduce and face all of these targets in order to have a rapid and detection. If you so you can see here what we have is what we want to achieve. The requirement for us is have pathogen detection, a multiplex pathogen analysis, drug-resistant detections, and also polymicrobial capability, which means if there are several pathogens in the same sample, we need to finalize and detection each one separately. And what we need to do in terms of operation in the hospital, in the clinic, and also in the point of care in rural area, we want to achieve a sample to answer, a rapid detection less than 1 hour, high sensitivity and specificity. Reliability is very important, and this is very scientific, as Prahad of the speaker talk, and also it must be integrated in the workflow. What we see here is our under development machine, which is already working as in a functional model that we have and several cartridges. What we see here is the NatLab machine with its computer interface and variety of cartridges in different color, which every color for us will mean a different panel of disease. So one color can reflect to sepsis, the other one tuberculosis, respiratory disease, meningitis, etc. And we will talk about the panel in the end. And our main feature, of course, are very important. If you will see in the next slide please, a true load and go system. We want this sample to answer disease. I will show you immediately in the cartridge. We want it to be rapid, under 45 minutes or 60 minutes. Minimum handling with this multi-parameter. We want to see many pathogen or distinctive pathogen in one sample, which will be reflect different pathogen of sepsis or other disease. It must be versatile with different clinical samples, scalable. We can use many units and connect them via IoT and random access and all this network system. So you can see in the next slide how it is working. from the clinical sample, put it directly to a cartridge, and then the cartridge will go directly to the NATLAB machine. And if you will see in the next slide, I will show you what is going on in the cartridge. So what you see here, this is the cartridge that we have designed and manufactured. If you will click one more, you will see where is the sample port to introduce the sample here. And on the next click, please, you will see the area of all the DNA or RNA extraction. So all the handling of the sample and the DNA extraction is happening inside the microfluidic cartridge. You will see the area of the amplification, the molecular amplification, which I will talk momentarily about. This is not a real-time PCR. This is isothermal amplification of the DNA, which happen rapid and specifically. You will see our unique IP carbon array electrode, where we can do more than 100 targets. So on each target here, on each spot that is very small here, there will be a different target for a different pathogen, so we can in parallel distinguish and identify one of many pathogens. What we are introducing in this cartridge, this is a different type of molecular amplification, not RT-PCR, but isothermal amplification, which we call, not we call, of course, it's in the literature, we call rolling circle amplification. The rolling circle amplification, it's a type of amplification adapted from bacteria where they amplify plasmid in isothermal temperature in a very rapid and enhanced manner. So in this technology, we are making much more simpler machines because we don't need a thermocycle and we need much less amplification. PCR amplification will take at least 45 cycles. Example, in here we can do the amplification in a manner of 10 minutes from the target. But what it is required, if you will see in step 1 here, it is required a circular probe, which is not fully circled like a plasmid, and it only ligate and be a full circle once it will recognize the DNA of the pathogen. So if it is hybridized on the correct area of the circular probe to the target pathogen, there will be an hybridization in the nicking area, and then there will be a ligation by an enzyme. Then only then, in step 2, after the ligation, we will see the full circle like a plasmid. And from this on, you can get an amplification like as I said, like bacteria amplify plasmid, and in 5 to 10 minutes, you get those steps of 4, 5, and 6 to have a very full amplification of the circular probe. And this circular probe contains the specific DNA, the diagnostic area of the pathogen. So if this is happening on every different pathogen, you will see only the circular probe that is specific to the pathogen that it will be amplified. We have adapted this all this amplification to be on a semi-solid RCA, as we call. We can coat magnetic beads in our system with different circular probe, and each bead, or sometimes several circular probe on the same bead, will be amplified only upon the introduction of the correct pathogen. And after this amplification of the RCA, we receive a very, very long double helix DNA, because we are doing also forward primer and reverse primer. I will not go into the details, but this we don't have the time for this. And after this, we are doing a cleavage of this DNA to a shorter, specific DNA. They are introduced to our carbon array, and only where the spot is specific to the DNA of the target of the bacteria that is existing in the sample, in this blood sample, for example, in sepsis patient, only then the relevant spot on the carbon array will be lit up with the fluorescent signal, and this is all happening inside the cartridge. So in here you can see, for example, a carbon array with 100 different electrodes. So we can do here up to 100 pathogen, different pathogen if we want. What we are doing in these cartridges, as I said, we are doing several different panels of disease. Of course, the one of the important is the sepsis panel, which include all those targets that are relevant to sepsis, bacteria, and also viral. We are doing other panels like respiratory panel, which include COVID, influenza, RSV, pneumonia, Legionella, and other. We are doing meningitis panel to distinguish meningitis disease between bacteria, viral, and fungal. We are doing hospital-acquired infectious disease panel for MRSA, vancomycin resistance, and other. We are doing very intensively now with the collaboration with the Stop TB tuberculosis panel that will include all type of different human tuberculosis, non-mycobacteria tuberculosis, and all the XDR antibiotic resistant of this. And we are doing other panel which may be less important, but these are all on our agenda. So just to conclude, the next slide, this please, this machine will be, we anticipated this to be launched by next year to 2023 with several important cartridges for several panel of disease. There will be a multi-respiratory panel, a meningitis panel, tuberculosis panel, and a sepsis panel. And we believe this will change the way of point-of-care diagnostic is happening today. And I hope to be next year in the meeting. I hope this will work, and I hope we can already show you for the next year our field experiment with those cartridges. So thank you very much, everybody.
Thank you, Eran. Okay, I think we've got one very quick question, if you could answer this. You showed a lot of diseases you're working in. Somebody is asking you, what type of samples can you use? Can it take whole blood?
Okay. We can take full blood in this cartridge. If you go back to the cartridge, a few slides back, in the sample port inside the cartridge, this we can take blood sample, we can take CSF, we can take BAL, we can take sputum after liquidification, and urine, of course. We can take all these kind of samples inside. Whole blood is absolutely welcome, as we say.
Thank you. Wonderful. So thank you very much, Eran. And we hope we do see you at that next meeting. So we'll move on to our next speaker. Sticking with microfluidics, we have Maïwenn Kersaudy-Kerhoas, who is a professor of microfluidic engineering at Heriot-Watt University in Edinburgh. Her group has been working on pre-analytic tools for extraction of circulating nucleic acids for various medical applications. And we're very delighted to have her here today talking on sepsis diagnosis with a cell-free DNA sequencing approach. So over to you, Maïwenn.
Thank you very much, Louise. So if we look at what is available in sepsis diagnostics right now, we have conventional solutions like blood cultures and complete blood counts, which are time-consuming protocols with poor diagnostic yields. On the other hand, we also have systemic infection biomarkers like PCT and CRP, which are specific and sensitive. They're not sepsis specific, and then they don't pinpoint to the pathogen responsible for sepsis. And finally, we have new entrants like Abioscope, for example, that can measure pancreatic stone protein, PSP, in a sensitive, specific, rapid way, and potentially also cost-effective. But again, this is not a solution capable of identifying the sepsis-causing pathogens. And at the moment, commercially, there is no solution that is able to do that and is also sensitive, specific, cost-effective, rapid, portable, and crucially allow targeted treatment. So in this presentation, I'm going to make the case for a microbial circulating DNA approach. Over 1,000 microorganisms are known to cause human disease, which makes the identification of causative pathogens difficult with current techniques. Presumptive approaches like molecular techniques are effective on a limited number of pathogens and could miss emerging pathogens. Now, what if? What if we could identify any pathogens in the blood circulation? A cell-free DNA approach could enable us to do just that. From seminal work started 10 years ago, we know that fragments of pathogens' DNA are released in host circulation. They are naturally fragmented and they are added to the host cell-free DNA population. It is possible to extract, to sequence, and to align these fragments to databases of thousands of known pathogens. Groups in the US, Blocamp, Shu, in Germany, Kaison, and in Japan, Yoshinori Ito, have already demonstrated the validity of such an approach. So now I'm going to look at this microbial cfDNA workflow, starting with sample processing. Good sample preparation is crucial for microbial cfDNA sequencing. In particular, host cells must be removed quickly to avoid further contamination of microbial cfDNA by host genomic DNA. So in traditional workflows, there's two ways to do that. You either do the manual bench extraction or you use robotic fluid handling platforms, which often also necessitate a number of manual steps, which leads to poor control of pre-analytical workflows and result variability, especially in multi-site studies where protocol variation between sites are an issue. And batch extraction also means variable and long turnaround time. Plus, you have to think about sample storage capabilities for storing blood or plasma in significant volumes. And it can be quite costly, especially in consumable equipment and experienced staff. Now, moving on to the sequencing part. A short-read sequencing approach is the most common sequencing technology for microbial cfDNA workflows. It has the advantage of high accuracy, but the disadvantage of fairly high capital investments, lack of portability, and fairly long sequencing runtime. So I'd like to point out at this stage that there is a cfDNA-based sepsis test that has been commercialized for the US market by a company called Kersaudy-Kerhoas, which retails at $2,000. Now, in conclusion, complex sample preparation and powerful but slightly cumbersome sequencing can hamper the timely clinical implementation of such a promising approach. I'd like to introduce our iSepSec workflow. In this project, we aim to develop an easily accessible, easily deployable, and cost-effective microbial cfDNA approach for sepsis diagnosis. Our workflow consists in 4 main steps, which we aim to deliver in under 6 hours. The first step is blood sample preparation. For this, we have developed our own custom cartridge-based cfDNA extraction automated solution, which provides an eluate of circulating cell-free DNA in just 45 minutes from plasma. Followed by this, we have a library preparation in 1 hour and 30 minutes. And after this, we aim to do real-time sequencing on an Oxford Nanopore platform in just 2 hours. And once we have the sequencing data, we use a bioinformatic pipeline, which is a free, online, cloud-based pipeline, which delivers a first report in just 30 minutes and a more consequent report in 3 hours. Now, what is different with our approach? Well, first of all, the timing is much quicker than current approaches for circulating nucleic acids. Then in sample processing, our cartridge-based extractions enables complete control, including immediate stabilization of cell-free DNA, immediate access to the material for clinical teams, portability, reduced storage needs, as you only need to store extract of cfDNA, and reduce cost. Now, in the sequencing part, we are using a long-read sequencing approach. The advantage there is the low capital investments, the real-time nature, in particularly of the base calling, which speeds up the analysis, the portability of the technology. The disadvantage is a lower accuracy compared to short-read sequencing. This is fast changing due to the fast-paced nature of the technology. A couple more things I like to say on our iSepsec workflow. It's completely culture-free and it's amplification-free. In terms of time to result, we easily beat the standard blood culture results. We also beat a commercial offer from Carius, which is 24 hours from the moment the samples are received. We can take up to 24 hours, so a total of 48 hours. So using this timing, most of which we already deliver right now, we can deliver a complete results in just 6 hours. Now, I'm going to share a few results that we've obtained on this workflow, starting with the cfDNA extraction. In the last 18 months, through optimization, we have stabilized elution volume to reach 60 microliters with a CV of 12%. And this has resulted in cfDNA recovery of an average of 77%, compared with the manual extraction technique, so the QIAGEN circulating nucleic acid, which is considered the gold standard in the industry, and which is known to be superior to existing robotic handling solutions. Next, we have applied our whole workflow to contrive sepsis samples. So the way we've put these samples together is using a fragmented microbial DNA mix containing 8 pathogenic species, 7 bacteria and 1 fungi into 1 milliliter of healthy plasma samples. And we've extracted 3 of these samples onto our automated platform and 3 samples on the bench for comparison. And then we have sequenced the eluates onto the Oxford Nanopore fungal device. So in terms of sequencing performance, we haven't seen any significant difference between the cartridge eluate and the bench manual eluates through 3 quality indicators, including number of reads, a Fred score, and percentage of unclassified reads. In terms of pathogen identification, we found similar detection patterns for the 8 organisms of this microbial DNA mix, which confirms the basic workflow capabilities. Now, bolstered by these promising results, we've acquired 10 septic patient samples from the Liverpool School of Tropical Medicine and the Malawi-Liverpool Wellcome Clinical Program. And all 10 samples have been extracted and sequenced, and we're still analyzing these results. But today, I want to give you a really quick peek into the type of results that we're getting. So in sample number 80 there, the sample came from a 20-year-old male, which presented at the hospital with fever, cough, chest pain, and difficulty breathing. The lactate reading was at 2.63, and the culture results notes only says no growth, but GPC seen. Now, using the iSepsec workflow, the organisms that we have identified in this sample is a Streptococcus pneumoniae. It gives a very strong signal at 8% of the total cfDNA reads, and this is highly consistent with the clinical presentation of the patients, which points out to the clinical validity of our approach. So in summary, we have made significant progress towards a technique that is sensitive, specific, cost-effective, multi-pathogens, rapid and portable for sepsis diagnosis. And it has the added bonus of having the capacity for epidemic surveillance and monitoring emerging threats. I'd like to thank the organizers for the invitation and the fantastic opportunity to speak today. I'd like to thank Anna and Linda at Hertford University, who did most of the technical work, Amanda, who were from the University of Edinburgh, who helped put the sequencing setup together, Kazuhiro Horeba at Nagoya University, who developed the PassDat, a cloud-based online and free bioinformatic tool for sepsis diagnosis, and Jamie Rylance and Chevin Jacob at the University of Liverpool, and Jacob Pulusa at the Malawi Liverpool Wellcome Trust, and everybody for listening. Thank you.
Thank you, Maïwenn. That was really interesting talk, and nice to see some of your data coming through now. I appreciate it. It's relatively early in development still, but there are several questions in the chat here, about what steps are being made towards making this affordable for lower resource settings where we've already discussed the burden of sepsis lies. Have you thought about that? I know you say it's cost effective, but how do you envisage it being used in these settings?
Yes, it's a little bit of a difficult question. It's cost effective compared to maybe the Illumina short-read sequencing approach that is commercialized at the moment, but it's not cost effective compared to some of the rapid diagnostic or even some of the established blood culture that we have at the moment. However, the first human genome costs £1 billion, right? So to sequence, and now a full genome takes just $1,000. So the scale of cost is just decreasing so fast that I hope that it will be affordable in the near future. There has already been some health economic studies on this that shows that it is cost effective to use such an approach, especially if it can diagnose specific causes like we've done in our pilot study, and which would avoid having, for example, a chest scan. So I hope that this gives some elements of answers to this.
Great, thank you. And then another probably one sentence answer. Again, the question is, what type of samples can you use whole blood?
Yes, so the automated platform itself only takes plasma at the moment, and our solution for the blood to plasma part is going to be a mini centrifuge that can take just 2 9 ml, so significant volume of samples. And I've actually published this. It's a preprint on bioRxiv, and hopefully very, very soon published in PLOS ONE.
Okay, just while we're getting the slides through to the next presentation ready, there's a question about health economic studies, actually, where can you point some of the audience towards where the health economic studies might be found?
Yes. If they go on the Carius website, in the publication section there, they should find health economic study that the Carius company and Timothy Blokam performed a few years ago. And otherwise, please feel free to contact me and I'll give you the details.
Okay, that's great. Thank you very much, Maïwenn.
Thank you.
We're going to move on now to our next speaker, Cecilia Ferreyra, who will be coming to us from FIND. For those of you who don't know about FIND, FIND is a global alliance for diagnostics. It seeks to ensure equitable access to reliable diagnosis around the world. Cecilia is an infectious disease specialist, but she is now also the director of the AMR program at FIND. She's going to be talking about developing a target product profile for sepsis diagnostic platforms applicable to high global burden settings. I think we can get going. Thanks, Cecilia.
Thank you very much, Louise, and thank you to the organizers and to the previous speakers for setting up the scene. I'm going to walk you through some of the initial thoughts that we are having at FINE regarding a development of a target product profile for neonatal sepsis. So I think we all are aware of the problem and the magnitude of the problem, and we know that sepsis is affecting millions of babies all around the world. And unfortunately, most of the mortalities and the incidence of sepsis is being seen in low and middle income countries. We also know that neonatal sepsis is the third leading cause of mortality. We know that the clinical algorithms that are being used today have a very low sensitivity and specificity, particularly because those have been done from a high income country perspective. We also know that broad spectrum antibiotics are increased adverse outcomes and are leading to antimicrobial resistance, which is going to be our concomitant problem when we talk about sepsis. The evidence on best diagnostics, and I'm glad to see some of the initiatives in these initial presentations and some technologies out there. But I think we need a bit more of an evidence-based diagnostics and recommendation for sepsis. And as we all know, and our first speaker already presented before, surveillance systems in LMICs are really poor, and we need more data on AMR and sepsis to better understand what we are talking about. And as you know, 85% of sepsis and around 85% of sepsis-related deaths are occurring in low and middle-income countries. So I think it's time to act now. What is the role of diagnostics for neonatal sepsis? And I think as an ID physician myself, I can say we don't know. We have an initial workflow. We do white blood cells counts. We use differential white blood cell counts, immature rates, platelets, glucose levels. But we know that none of this is the gold standard. We have to use a combination of diagnostic solutions that are not always giving us a fast enough response to treat the babies. And also the time error results, we all know is a big issue. Blood cultures take days. We are seeing some near patient point of care technologies, but are these the ones that we should be using? And babies pose a particular problem in terms of volumes. How much volumes of blood are we needed to do all this menu of diagnostics technologies? So what we know until now, and that's why we are scoping a neonatal sepsis TPP for neonates, is that there is a lack of clear definitions on neonatal sepsis, both clinical and diagnostics pathway. The different etiologies between high-income countries and low-middle-income countries make it quite challenging in terms of defining what will be the best panel for diagnostic approaches as well. We know that blood culture has been the gold standard, but with a low performance. We know the challenges to get enough volume from the babies, but also the positivity rate and contamination rates are very low and high in this particular population. Biomarkers, there is a lot out there, but there is nothing out there in terms of enough evidence. What is the right biomarker that is useful for this population? There is also a lack of evidence on the role of other commonly used diagnostics like glucose, fluid blood cultures, lactates, and moreover, most of these tests are not widely available in LMICs, which makes signs and some symptoms have been used in the clinical algorithms to identify cases. Also, we know that in LMICs, the cost and access to tools is one of the main challenges. We've seen that for COVID, access to testing and vaccines has been really poor, and the same is applicable for sepsis. jump one. So at FIND, we start looking for what is out there, what is the technology out there, and this is work in progress that I hope is going to be published soon. So we've done a technology landscape search, including different company websites, conferences, articles, literature reviews. We searched for sepsis, test technologies, neonatal sepsis. We did not exclude complex technologies because there are so few technologies on the pipeline that we didn't want to limit the search. So we haven't made a wide explore of technologies out there. Many companies, for example, offer a single-plex lateral flow test for a single biomarker like CRP or PCT, but we also focus on other platforms that will integrate more biomarkers relevant to neonatal sepsis. And we developed different scoring criteria, whether it has a neonate application or a sepsis-related assay. So what we find out until now so far is biomarkers for sepsis. You know that there are many studies ongoing to define which biomarkers are performing better in this particular population. But again, very few studies have been done in low and middle income countries, and very few studies are ongoing right now. The only biomarkers what we find out with a high sensitivity and specificity has been cell surface markers, both CD11b and CD64, but we know that other biomarkers like ELCs or ELA have shown good performance in high-income countries. And we also know the poor role of CRP and PCT, but we also know that they do have a role, maybe not to rule in sepsis, but to rule out or to reduce antimicrobial consumption. So there is a lot of evidence needed in this field from those settings. But then again, what some considerations for use in these settings? What is the equipment required for detection? Can ELISA, a flow cytometry, a lateral flow test? What is the right format? Knowing that in most LMICs, power supply is an issue, lack of lab infrastructure, but also lack of trained HR. So we had to make it easier, but at the same time, we want a quality and a good performance. Does the biomarker require quantitation or qualitative information will be enough? We need to better understand the time frame for biomarkers increase and decrease in neonates. We know it's pretty challenging, the nature. Are multiple biomarkers required? Like maybe a combination of different biomarkers will increase sensitivity and specificity? We don't know. So we need more data from LMICs. We also find out other technologies for sepsis. Some of them are multiplexing. Some of them are very early stages yet. Some of them are robust enough for LMICs, others are not. Others requires a more sophisticated laboratory. The sample prep is a big issue, as I mentioned before. We need to work with small volumes of blood, and the ideal will be that with that small volume, you can have all what you need, including glucose and white blood cell counts. We are following up on some new technologies that are out there. Some of them are targeting LMICs, some others are not, but they will be interested on those, some evaluations in those settings. And I want to frame here what Fraley and colleagues wrote in this paper, which I found it really useful. How an ideal diagnostic test for sepsis should be. We go again into the ideal that we are always asking for, has to be rapid. It has to be a broad-based detection, should include bacteria, viruses, and fungi. It should be minimally invasive. We know that the volumes of samples are really critical for these babies. It needs to have high sensitivity and specificity because it needs to be better than what we are using right now. It should have a polymicrobial detection because we know that contaminants is an issue. It should include drug resistance because this is coming, this is happening already. It needs to be integrated into the clinical workflow, so it has to be easy to use and require a minimal expertise. We know that in most of these settings, just clinicians are running the whole show. It should detect unknown and emerging pathogens ideally, and I think that COVID has shown us how important it is to have multiplex platforms, and it has to distinguish the inflammatory response in either host or pathogen driven. So what are we doing right now is drafting a TPP, and why TPPs are important, particularly for product development partners like FIND, because these are the key to use manufacturers and guide the manufacturers. And if we have a poor understanding of what is the clinical need, the results are really, is that a test will be poorly used, we are going to have tests that are not fit for purpose and will never reach the market. Also, the TPPs help to provide a clear guidance on how the test should look like, should work like, what the price should be. So it's a whole combination of information that is useful from beginning to the end of a test development. So where we are now is on the phase 1, scoping the TPP. So as I mentioned before, we are already doing a landscape analysis of what is out there. We are doing a literature review of available data. We are already having meetings with key stakeholders and other key opinion leaders to define what will be the best test characteristics and the priority setting. In 2020, I think, this report from UNICEF and NIC-NEST 360 has been published. They were intending to have a TPP for neonatal sepsis. And as you can see, they developed six use cases, and that showed the complexity of neonatal sepsis. So the difference between most of them is whether to start antibiotics or to stop antibiotics, and at which level of the health system the babies are seeing. And of course, there are technology considerations at each level and for each use case. So what are the next steps from our side is also link it with neonatal sepsis, but more broadly targeting bloodstream infections and severe patients. We are supporting the development of a simplified blood culture system that should be able to be sitting in a level 2 facility in a low and middle income country. So it's not going to be a huge, massive MALDI-TOF-like. It's going to be smaller and more simpler to use technology. And we are also working on developing a molecular panel for pathogen ID and ESD, because we know that until the sequencing and the new technologies arrive, something has to be done in these settings, and to get simpler, that will be the best way to go. We are going to define also, we are going to work with experts to simplify the use cases that I mentioned before, to develop the TPP. We are going to prioritize the needs. What is that we need? What is that clinicians need in LMICs? Do they need enough information to start antibiotics, to stop antibiotics, to track severity? What is right now needed? And from there, we can start developing the next steps. We need more evidence on biomarkers and the combination of them. What works better, or it doesn't work, and in which kind of platform? Is a lateral flow assay better than a multiplex? So we are going to be inviting key stakeholders to be involved in this TPP definition, open an expression of interest for different manufacturers to start the discussions, and at the same time, that's something that we always do at FIND, is we start having an engaging with countries discussion to be prepared for the further rollout, because you may develop the best technology in the world, but if this is not finally accessible to LMICs, I don't think you are successfully achieving what you aim for. And with this, I want to thank all of you for listening and all my colleagues supporting this work. Thank you very much.
Thank you very much, Cecilia. That was really interesting. And I think you brought up many of the challenges faced all over the world, not just in LMICs in diagnosis of sepsis. I have a question for you, really. We're talking about LMICs in general, but we all know they're highly variable, and the causes of sepsis are very variable, even within countries, let alone throughout the world. Have you considered that, and how will you integrate that into your target product profile?
Yeah, that's a great question, and you are right. Even LMICs have a difference. A neonatal intensive care unit in South Africa is not the same that in DRC, and we are really aware of that, and that's why when you define a TPP, you are having different key stakeholders sitting with you and defining that TPP, and then you finally reach to an agreement. And I have to say that basically the more simple tool that is useful for that very poor resource setting will be definitely useful for a better suited lab. So I think that finding the balance of what works best for both, that's what a TPP aims for as well. And the TPP finally goes through a selection criteria with voting. So there is a lot of statistics behind, and there should be an agreement within what will be the best for one setting versus another one.
Thank you. I don't want to encroach too much, perhaps, on the next speaker's talk, but there's a lot of chat about making these type of tests affordable in lower resource settings. And can you give us some examples of how you approach this? I mean, obviously, it's very important to the project, but how you actually go about doing this?
Mm-hmm. Great. So I'm happy to see that our Diatropics colleagues are there. So I think that's going to be also some useful information on how you can shape the market for LMICs. And I think that something that I find always tough when developing any test is you start having discussions with those countries from the day zero. And also the discussions we have with the manufacturers are pretty clear in terms of the target price, and those target price are also included in the TPP. And we know sometimes TPPs can ask for $1 price, which is not feasible, and we are aware of that. So we are trying also with the price in the TPP to make a good balance between what is the real cost of goods of the technology, what will be the benefit for the manufacturer, but basically what you need is to make this technology accessible. And I think a lot of work has been done on COVID at FINE through the ACTAID accelerator to bring down the prices of the rapid test for COVID for LMICs. We've done it also for other technologies for TB, malaria, and NTDs, for example. So that's our core work as well, and also making sure that the manufacturers, when they apply, they are pretty aware of our global access policies and prices.
Wonderful. Thank you. Thank you very much, Cecilia. So we will now move on to our final speaker for this session. And it is my pleasure to welcome Cheikh Tidiane Diagne. Cheikh originally trained in Toulouse in France with a PhD in structural and functional biology, but he now leads a Diatropics laboratory. For those of you unfamiliar, Diatropics is a social venture. It's founded by the Pasteur Institute in Dakar, Senegal, the Merieux Foundation, the Foundation for Innovative New Diagnostics in Geneva, and the French Institute for Research and Development. Diatropics focuses on local manufacturing of rapid diagnostic tests for neglected and epidemic diseases in Africa. So over to Cheikh, the floor is yours.
Thank you very much, Louise. It's a privilege to talk about accessible and affordable scale-up of diagnostics in Africa. LMICs have limited access to healthcare, and the Institute for Health Metrics and Evaluation showed that the countries with the lowest GDP per capita share the highest burden of communicable disease. And most of these countries are based in Africa. And such burden has tremendous impacts in terms of health, but also economical and social impact. Diatropics initiative started before COVID-19 in order to bring diagnosis capacity for infectious and neglected disease in Africa, and to do so for visionary partners, which are the Merieux Foundation, FINE, Institut Pasteur de Dakar et Institut de Recherche and Development successfully partnered to deliver one single goal, which is the promotion of access to diagnostics for local manufacturing, but also by setting up an innovative access model. Diatropics will manufacture tests in 3 different scenarios. The first one is when the tests are not available. Second one is when the tests are available but not satisfactory in terms of performance, because sometimes tests can work very well in a country, in European country, and not work in Africa. And the third scenario is when the tests are available but not affordable for the African market. Diatropics will leverage on the lateral flow technology, and will mainly work through industrial partnerships. So far, 3 technical partners have joined us. They are Monogic in the UK, Bionote in South Korea, and Biomerey in France. Manufacturing at Diatropics is very demand-driven, and the demand can come from different parties. In order to ensure an affordable supply, the parties will pay a membership fee, which is a yearly subscription that covers fixed costs. And hence, they will have access to the Diatropics test pipeline, and will be charged only the manufacturing fees, which are more or less equal to the cost of goods. This allows Diatropics to have a price tag for its members, and also work efficiently with different leaders in health in Africa, like the Africa CDC, and also the West Africa Health Organization that covers the 15 countries in the ECOWAS region. And such model also allows us to avoid to do the distribution via a third party, because that will add a margin to the final cost. Basically, the price cut offered by Diatropics through the mutualization with the membership covers 60% of the cost of an RDT, and these are the operating margin and the labor costs. And the price charged to the Diatropics members will cover 40% of the cost of the RDT, which are the facility maintenance, equipment, cost of goods, but also costs related to quality. As I was saying earlier, Diatropics has started before the pandemic. It started on September 2019 with the construction, and it rapidly ramped up with the operations and the manufacturing of the first COVID-19 antibody. And on November 2020, Diatropics has been ISO 13485 certified by the British Standard Institutions. And as of March 2021, 4 members have joined Diatropics. They are the Open Society Foundation, Médecins Sans Frontières, the West Africa Health Organization, and the Africa CDC. On July 2021, the first market authorization for the COVID-19 antigen was achieved in Senegal. And on October 2021, the first regional emergency use for the site to COVID-19 antigen. And on December 2021, Diatropics has been allowed the Ghanaian prize in the category of the best medical technology for its COVID-19 antigen test that has been distributed in Africa. In terms of manufacturing processes, there is a quality control, formulation, treatment, assembly, and packaging. And all of these processes are run through the ISO 13485 standards. And the Diatropics is in a capacity to transfer technology from anywhere, any steps of these processes. It means that it can receive raw materials and go through the quality control of the raw materials to the packaging and the final batch release, or it can also receive the semi-finished products in order to ramp up quickly this capacity. And this is what has been done for the COVID-19 antigen. In the pipeline, currently there are COVID-19 antigen and antibody tests with Bionote and Monogic as technical partners and with different funders. But also there is hepatitis B antigen, the meningococcal meningitis, yellow fever antigen, dengue antibody and antigen, measles, rubella, Ebola, the African trypanosomiasis, and also the Marburg. Marburg has been recently funded by FINE, and it's the only test for which there won't be a technological transfer. Diatropics will be doing the R&D. To go quickly on the COVID-19 product that have been manufactured so far, there is the COVID triple antibody COVID-19 test. And this detects IgA, IgM, and IgG from blood samples. And the global sensitivity for IgA is 12.1% and specificity 100%. For IgM is 92.5% and the specificity is 91.9%. And for IgG, 67.8% and specificity is 100%. So the performance for IgA is very low. It's because IgA blue sample is not the correct samples to detect IgA, but we have noticed here at hospitals that IgA might be a good biomarker for severe COVID-19 cases. And when we look at the performance, the sensitivity per days post onset of symptoms, the maximum sensitivity for IgA is reached beyond 21 days post onset of symptoms with 17.02%. And for IgM, maximum sensitivity is reached at between 7 and 14 days post onset of symptoms, and it's 96.36%. And for IgG, we reach 83% beyond 21 days post onset of symptoms. For the COVID-19 antigen test, the clinical sensitivity is 89.9%, and it can reach 92.5% when we look at days onset of symptoms below 7 days, and 97.2% if you look at sensitivity inferior or equal to 33, and 100% when we look at sensitivity for CT value below 25. And overall specificity is 98.6%. Currently, there is a manufacturing capacity between 2 and 4 million tests per year, so which is quite small. But thanks to the ACTE initiative, we have been funded by FIND and UNITED to increase our capacity and beyond 50 million tests a year. And we believe that this capacity is a strong support to the PACT initiative set up by the Africa CDC, which is a partnership to accelerate COVID-19 testing in Africa. And as of February 2022, less than 100% COVID-19 tests have been used in Africa, and we believe this capacity can play a strong role in testing in Africa. And also, we are ramping up our capacity, manufacturing capacity. Diagnostics needs to broaden its activities and find a niche, diagnostics niche. And we believe that niche will be between what anti-pathological Dakar question, but so profession, vocation, and mission. And this will certainly be at the intersection between what we love to do, which is serving Africa and promoting health. So what we are good at, which is project development, technological transfer, so industrialization for tests, but also as previously demonstrated with the yellow fever manufacturing at IPD. So what we can be paid for is offering manufacturing capacity, but also offering technical expertise, franchising the branding, and so brand segmentation. And finally, what the world needs, I would say mainly Africa, what mainly Africa needs, which is access to high quality diagnostics and local manufacturing. Thank you very much.
Thank you, Cheikh. That was really, really interesting and followed very nicely from Cecilia's talk. I guess a question from me, following really from your last slide, would be, from your point of view, what do you think the priorities are, and in terms of what you love and what way you would like to go, where would you move into in terms of diagnostics, assuming the others factors would align with that?
Thank you. That's a good question. Actually, we are thinking about going through AMR testing, and I think sepsis one will be a very good candidate. That we're thinking about not only focusing on communicable disease, but also in non-communicable diseases such as cancer-related disease.
Okay, thank you. And then a couple of questions coming up in the audience chat. A fairly specific question about the sensitivity and specificity, I think, of the COVID tests, from which regions of Africa and where were people involved, or what were the populations used in this validation?
So for the COVID-19 antibody tests, it has been tested in Senegalese hospitals here in Dakar. It has been tested on COVID-19 suspect cases. And for the COVID-19 antigen test, so this is a OEM OBL product. It has been independently evaluated by FIND DX. We also did same evaluation here in Dakar in the same hospitals where the COVID-19 antibody test has been done on COVID-19 suspect cases.
Wonderful. And there is a question more generally about common pathogens, and perhaps I could spin that round to, in terms of developing diagnostics in the future, are there certain pathogens that you're thinking about already in addition to the COVID-19 work?
Yes, so in the pipeline, so there are a couple of tests on which we are focusing on, which are the hepatitis B, meningococcal meningitis, yellow fever, dengue, measles, rubella, Ebola, African trypanosomizin, also Marburg. And for meningococcal meningitis, we are thinking about having a new test that will be focused on Listeria, monocytogen, also on E. coli and Haemophilus influenzae. These are the tests that we'll be focusing in the next 2 years. And if there are some partners like FINE or other parties, we'll be happy to tech transfer any test that will be of use for the African populations.
Wonderful. Well, thank you very much, Cheikh. And your talk brings us to the end of this first session of today's meeting. I would like to thank all of our speakers in this session. Some wonderful talks and very thought-provoking. There's a lot of chat in the box, and I hope that the speakers will be able to join the rest of the meeting. We will follow on after a short break with our next session, which will start on the hour, so in about 20 minutes time, I think. On biomarkers in the diagnosis and management of sepsis and COVID-19. I'd like to thank all of the sponsors who make this meeting possible. They enable it to be free for people wherever they are in the world to join. And I really would like to extend our thanks and gratitude to the sponsors. And finally, to thank all of you participants in the Congress as well. You're essential for the meeting, and it's amazing to see people from all over the world and all times of day and night joining in the chat. These sessions will be available starting on the 3rd of May, available on Apple Podcasts and YouTube, so keep a lookout for those as well. But with that, I would like to bring this first session to a close. Thank you very much.